Okay, so I brought you here today to talk about a new technology that is revolutionizing medicine. This technology is called CRISPR-Cas9. Now, what is CRISPR-Cas9? CRISPR stands for Specifically Clustered, Regularly Interspaced, Short Palindromic Repeats Technology. And this technology is a gene editing tool where scientists can use it to research treatments for the disease, they can research cures for the disease, and they can also help prevent diseases in the human embryonic stage. Now, this technology is a huge breakthrough for biologists. For years, scientists have been trying to find a way to stop disease, and now they think they've found it. Now, how does CRISPR-Cas9 work? Well, CRISPR uses a guide RNA. This guide RNA is a specific sequence of DNA that looks for other damaged sequences of DNA. Once the guide RNA finds the damaged DNA sequence, then the enzyme Cas9 jumps in. An enzyme is a biological catalyst, which means it's used to speed up or slow down processes within the body. The enzyme Cas9 sticks onto the damaged DNA and begins splicing the DNA, which means to cut. Once the enzyme has cut the DNA, then the inserted fixed DNA sequence is placed into the original DNA strand. This fixed DNA sequence is something that scientists have researched that will be used for treating or curing that specific disease. Now, this tool is being used for multiple purposes around the globe. Now, how did CRISPR-Cas9 come about? So if you think that's cool, just wait until you hear this. In the late 50s, scientists just discovered the molecular makeup of DNA. A couple of years after that, a researcher named Ingram examined a specific chemical alteration in a hemoglobin protein, and he concluded that this alteration was the cause of sickle cell disease. Now, this was the first discovery that diseases could be linked to our genes. About 10 years ago, humanity discovered that we could check for genetic diseases in human embryo, which gave humanity the option to abort the fetus or not. But now, humanity can simply erase the diseases from future populations using CRISPR technology. Okay? And what else can CRISPR do? So there's a couple things that Dr. Zhang has been looking at to study for possible future directions for CRISPR-Cas9. One of the things that he has found to be is that he can use CRISPR-Cas9 to reverse a percentage of blindness in animals. Now, he uses the gene that causes the blindness, and he manipulates that genetic code so that it reverses the blindness. Now, in the diagram I have pictured below, the big purple blob is our Cas9, and the green DNA sequence is our guide RNA. The guide RNA is set out to find the incorrect DNA sequence, which is where the scissors are pointing to. Once the scissors cut out the DNA sequence, we are left with just the mouse's DNA and an empty genetic code where he places the repaired DNA sequence into the genetic code and then re-puts that into the mouse. Now, he has been using this technology for the last 7 years and has found out that any blindness that is caused from a genetic disorder can be somewhat reversed in all animals. Okay. So, in conclusion, what can CRISPR actually do? So for one, CRISPR can cut into any DNA and change the flawed DNA sequence, which is most likely the cause for the disease in the patient. This technology can be used for treatments for those who have already been born. So if you have been born with the disease, this technology is used as a treatment. But if you are in the human embryonic stage, this technology can be used to erase the disease out of your DNA. So the human embryo will no longer be born with the genetic disorder. So what does this do for humanity? Well, it saves a lot of money and pain, and you ask how? Well, for years, people have been praying for a technology that can save their loved ones from leukemia or muscular dystrophy. But now we can. This possibility can save a family from raising a child with a rare, high-maintenance genetic disorder, saving that family tears and pain from seeing their loved one in pain or suffering. But it doesn't just do that. It also saves that money, saves that family the money that they would be using to treat that genetic disorder for their loved one's lifetime. So in all, this technology is a huge breakthrough for scientists, and hopefully we can go on to curing not most, but all diseases in the human genome.